I'll have to, yes, here we go.
Take it away, Vince.
Got it. All right. Well, thank you very much, folks, for coming on board here. You're going to see 12.5 cents worth of computation if I talk for half an hour. You see the rate there, very inexpensive session we have going here. So thank you for coming. I'm going to be talking about bioconductor in AnVIL with a number of examples. And the examples are going to involve very improvised computations assisted by AnVIL and AnVIL assisted by bioconductor. And I think there's always been a bit of vagueness about the relationship between bioconductor and AnVIL, and I'm not going to be able to solve that here. I consider them to be quite symbiotic. And there's always this issue of, is it RStudio? I mean, AnVIL gives you RStudio, and basically you can get anything in CRAN, you can get bioconductor and so forth. So really RStudio is the gift that AnVIL gives to the data analyst. And there's many senses in which that's true. It's not the easiest thing in the world to get a computer, a nice big computer that has RStudio running easily for you. And the folks at the AnVIL and the Terra Tech Group have really accomplished that in a very nice way. And we've been working on the AnVIL for quite a while. And I think early on, we recognized that there was a tremendous capability of getting cloud computation in the framework in which we can do data analysis in a familiar way with R and RStudio. Let's see how that's done. When you get yourself into an AnVIL workspace, you have the ability to configure a cloud environment. And the one I configured here is the RStudio Bioconductor cloud environment. And hope I don't mess it up. But you can see here there are application configurations, and I've picked the RStudio one, which is a community-maintained RStudio environment. That means that the Bioconductor folks have been involved in producing the container within which all these computations are going to be conducted. And that's mostly the work now of Alex Mahmoud, who is in my group at the Channing Division of Network Medicine. So you can see here that you get RStudio with R 4.3.0, Bioconductor version 3.17, that's the current release, and a nice flavor of Python. You can check what's installed on this environment and see that there are many Python packages, and then if you check the R packages, you can see that there's a great many packages that are pre-installed. I will be using some packages that are not in this list, and that is one of the great virtues of Alex's work on this, because these are, and almost any package you might need from Bioconductor or from CRAN is available as a precompiled binary, and it installs very rapidly. Never been here before. Let's hope I can get out. This is the RStudio cloud environment. We'll just X out of there. And now here we are in RStudio. And it may not look so good because I wanted to shift over here, but with RStudio, you have a lovely console and many different tabs here to manage what you're going to work on. And what I would like to start is by looking at the new project window. And once you start working with a new project window, it asks you what you want to do with your current work. And I think I'm not going to get too involved with that right now, but I'll just mention that there is effective, look at that. I must have clicked the wrong thing. Oh, well. Let's not get involved with this. Is it possible to cancel this? Yes, good. Let's do that. You can get an interface to Git. And if you have GitHub and Git repositories and so forth, it's easy to make a Git managed or GitHub managed source code repository that interfaces very well with RStudio and therefore interfaces very well with the AnVIL. I'm not going to get too deeply into that right now. If you have questions about that, we can talk about it later and maybe even get some Git activity fired up. But it's definitely a good way to get engaged with AnVIL and manage your code. So one of the things that I did with Git and AnVIL and so forth was to install a new package that I'm working on called GWAS cat search. And I won't go into the details of how you get it installed. It's in my own repository. But if I start GWAS cat search attach it, load it as a library, I get access to a bunch of functions. And let's just hope that that goes very quickly. It does. And one of the nice functions it has is called search GWAS cat. And what it does is it fires up, and I hope you can see this, a Shiny app. And this was one of our big joys when we started working with Bioconductor and AnVIL, that it was possible to write new interactive apps that could run through the RStudio interface. And this one is something which is engaged in the topic of allowing semantic search of phenotypes that have been cataloged in the GWAS catalog, and attempting to map those, the phrases that investigators use to describe the phenotypes which are the outcomes of their GWAS to formal ontology tags. And here we're using the experimental factor ontology. And we get a list based on this query here, which is a semantic query involving possible wildcards and phrase logic. So we can try different phrases here. The one that comes sort of bundled in has to do with kidney disease. And we have the option of clicking on one of these, or we can just find all of the resources, all of the GWAS that are cataloged for diseases that are, or measurements that are related to these semantic terms. And when we've done this, we can pick one of these, or we can visit the PubMed reference for this. What I wanted to do was click on one of these studies and then click on the SNPs tab. And it shows me which SNP was cataloged by the EBI GWAS catalogers as related to the phenotype chronic kidney disease. And if I click on that and then look for the Manhattan plot, I'll get a very nice display, not only of the Manhattan plot for this study, but for any GWAS that has been cataloged that has a SNP in the vicinity of the hit for that selected SNP. So you can see here, the top SNPs have to do with hemoglobin. The SNP that I was actually looking for based on chronic kidney disease is down here. So it's in the vicinity of a great many GWAS hits. And the hope with this tool, which is just in its very early phases, is that this will help folks both identify phenotypes that have only been informally characterized, identify them in an ontologically meaningful way. So here we're linking measurements like urate or GFR changes to chronic kidney disease because they are, both annotated in the experimental factor ontology. We found a SNP, and we looked at other GWAS hits in the vicinity of this SNP, which is here. So this is a way of using AnVIL to introduce new software that works with R and Bioconductor to help people do genetics of human phenotypes and human diseases. So having consumed about 10 minutes, I will pause to see if there are any questions about this. To unpack this a little bit more, we can look at what is involved with our search list here. So in order to run the GWAS catalog search, we have to use multiple packages, and the ones that have been directly attached are here. And R and Bioconductor arrange that you get all of this software here. If we look at the second element of my search list, we can see that there are many functions involved here. And the ones we are using are make current GWAS cat, for example, to get the current GWAS catalog. And then we're also using G ranges to find the SNPs that I have plotted for you just a moment ago. So there's lots of Bioconductor infrastructure here that was taken advantage of to do this display. All right, well, that is one way of using AnVIL and Bioconductor together to do some research. The next thing I wanted to look at has to do with the Human Pangenome Reference Consortium, HPRC. And there's a very nice dashboard that was composed mainly by the folks who set up this workspace, the HPRC. And we'll get past this little bit here, but this is how the HPRC describes its own workspace towards a complete reference of human genome diversity. And there's wonderful papers published about this. Maybe we'll take a look at them, look at one of them later. And the tables that have been prepared by the HPRC in this workspace are what we're going to get into as we work with this workspace. So the interesting thing about it, or one of the many interesting things about it, is that we don't really know how to do the pangenome. There's a hope that you can get very clear information on cohort level or individual level variants, complex variants that are not SNPs and are not simple indels. But how exactly to do that with all the repetition and other technical problems with current genome references is an open question. And so there's lots of long read sequencing techniques that have been used here, and there's all kinds of benchmarking going on to try and help people get a sense of what the best approach will be to produce a pangenome that really reflects human genetic variation at its most comprehensive level. And so, this is a really nice self-describing resource about the Pangenome project. And the question is, how can we use Bioconductor to help navigate all of this? And I hardly know, I hardly remember, and I have to scroll around here. I seem to have lost, oh yes, okay. So this is all this wonderful prose about what went on in this particular version 1 of the Pangenome project. These are certain quality control metrics for the 47 samples that are being used. And we're going to look at the geographic layout of these 47 samples in a moment, if everything works out well. So the genome graphs, that's a new concept for me, ways of representing the pangenome so that you have, a lot of linear sequence, which is shared by everyone, and then bubbles where different types of complex variation are observed. And again, this is an extremely rich resource here. You can click on these links and find out all about the different algorithms that are used to represent pangenomes. But what can we compute? So let's get going. Let's go back to our RStudio session. How do you get back there? You have to click over here and then open, and never push the pause button unless you want to have many minutes of delay. Hopefully I didn't do that. And we are back in RStudio. Here we are. So let's think, what did we do in order to work with the pangenome reference? And what we did is we created our own package. And let's see what files are in the package. Wish I had a mouse. Scroll around a little bit more easily. Let's turn it on and see if we can get it activated here. Yeah, there we go. So here we are. Aha. Okay. So I made a package, and a package has a description file and a namespace and some folders associated with it. Let's have a quick look at the description. Here it is. And that pops up over here. Let's make our window a little bit bigger, if we can. There we go. And then pull this down a little bit. And look at the Human Pangenome Research Consortium resources, version 0.0.3. I identify myself. I have a bug report place. It uses all kinds of packages. And I think what I want to jump to here is the use of the AnVIL package. And I'm going to run a function called AV tables. This is one of the most important functions that Bioconductor has contributed to the AnVIL. because it lets you work with workspaces in a very rational and programmatic way. And it knows all about different features of your authentication and so forth. And this is really tremendous in my view. This is how I started to learn about the pangenome at all. So there are 7 tables here with very nice names like minigraph. So there's actually some minigraph outputs, algorithm run over the 45, 47 or so samples from which the pangenome reference is being constructed. And PGGB, assembly sample, participant, and so forth. These are all tables, and they all have their own column names and their own number of rows. Well, it's a little cumbersome to look at these things. So I wrote a little app, Grab Tabs. And it's very specialized just for this workspace. But now it's allowing me to look at these tables and get a sense for what's in there without poking around our programs and tables and codes inside the console. And I'm not going to claim that it's easy to look at this, but you can see that there's a lot of Google storage being used, and maybe it's a bed file or a VCF or a BAM file, and that's really wonderful to know because I can then use programs to go and dig into those files when I resolve these Google storage references. One of the nice ones is participant, because it has information about sex and the population of origin of the different samples. I wish it would say a little bit more about the actual cell lines that are being used for this, but this is fine. You can probably dig into some of the other references around to find out exactly what's going on with these, the biology of extracting complex variants from the genomes underlying these samples. So that's all wonderful. Let's see what a minigraph looks like. The minigraph table here has only 2 rows, but it has an interesting content. 3 different things: a GFA, which is a graph representation of the minigraph for the pangenome, and some bed files. And I know how to do things with bed files, and that's what I did. I haven't looked at the GFA yet, but that will happen. But let's work with the bed files, and actually let's make some G ranges and figure out what's going on with these bed files. If you read them, you see this one is minigraph CHM13. And this one, these are the TBIs. This row corresponds to CHM13. That's the T to T reference genome. And this one is GRCH38, build 38. I know something about that. I know something about this too. In fact, Bioconductor has a number of resources. It certainly has the full linear sequence of the T to T genome. And I think it's the current version. I also made a transcript library for the T to T genome, but I don't think we put that in Bioconductor yet. GRCH, we have plenty of stuff on, GRCH38. So that's nice. We have an app that we can use to investigate different aspects of the pangenome reference. Good. Well, let's close that app. And now let's go and look at the data that we've already assembled inside the package equals BioC, HPRC, LK, which just means it's a look at the at this thing. And here's the list of the data sets. So let's go and grab this mini GR GRCH38. Data. No. Somehow RStudio is a little too helpful. And what is this thing? Let's blow this up a bit. Get a nice big view. Mini GR. One of the things I was inspired by here is a very nice video in this series of AnVIL demos that was given in April, I think, by Julian who actually, constructed these pangenomes or has worked on that. So it's a half-hour video where you get a lot of information about the underpinnings of these computations. So I would definitely check that out. This is what we made. This is a lossless representation of the bed file for the GRCH38 mapped minigraph for the HPRC version 1. And it's a G-ranges. It's a G-ranges with some metadata. I thought it'd be interesting to look at the representation of these longest paths through these bubbles that represent the complex variants. And if you just say here, longest path, you get something that's a little more vivid, I think, in terms of telling us what it means that we have these complex variants there. And in certain places, I think if you look at one of the other representations here, you can see a lot of repetition between these complex variants in certain places. So this is the work of Hervé Page, the representation of DNA string sets with the coloring that helps you figure out whether there is structure in some of these variants denoted as not nucleotide sequences. So that's pretty nice. And we have the minigraph of both types, the GRCH38 and the T2T, I think, in our data here. Let's just verify that. We'll go back to our shared approach. Yeah, mini GRCHM13. So you can have a look at that. To wrap this up, I am going to run one more function that I just wrote, which is a kind of sanity check of the HPRC data that we've got. So, a long story short, there's a gene that's related to hepatocellular carcinoma called SIMD3 or SMYD3. I think it is, yeah. And what I did was I took a VCF representation that's available in the HPRC and took a little segment of it around this gene. Why did I do that? Because I sorted the complex variants. I sorted the genes, sorted them by gene, and counted the number of complex variants annotated into the region of every gene. You can do that easily in Bioconductor. I'll describe how to do it in this package. And SMYD3 had a lot of complex variants nearby. So I wanted to understand something about that, and so I made this VCF excerpt. So let's get that data. Come on now, type for me. SMYD3V. So this is a classic structure for VCF data made many years ago by a couple of folks in Bioconductor. They've gone, so maintaining that software is a bit of a drag, but I think it's working okay. This is what we get. This is called a collapsed VCF. And it's a very full representation of the information in the VCF that was created in the HPRC. And you can see that for this region, which is around this gene, I picked up 20,747 variants on 45 individuals. So there were some quality issues that led to the exclusion of some of the samples, but that's all upstream. We're just working with the genotype data here. And I wanted to check that we could recapitulate what we know about the population origins of these contributors. And that's a function called simple PCs, which is in this package here. And we'll look at the code, we'll run it, and then we will stop. Not found. Maybe it's like that. No. Sorry, folks. We do have problems every now and again. Looks like I didn't. Yes, I didn't library. BioC HPRC. Okay. Getting there. Okay. Let's see. There it is. So let's run it. in order to run it, I need some data. I have the SMYD. We have that. And what else do we need? We need the participant data. So data, actually, it can be done very simply. Just say example, simple PCs. Okay. Let's see what happens. Great. We got a plot somewhere. Okay, this is excellent. So we ran the example, which is a nice feature of making your code into R packages. And yeah, this is a display of the variation, only the biallelic SNPs. So we got rid of all the other ones because we don't really know what to do with them in this context at this point. But there's a nice package called SNPstats that'll do that for you. And it'll give you only the biallelic SNPs, and then you can do a principal components analysis on the numerical representation of those genotypes. And what I see here is somewhat comforting. It's a very small amount of the genome, but it still shows reasonable sorting of contributors in terms of groups that seem to have common geographic origin. So with that, I will close and invite your questions. Thank you so much for your attention, and I hope this was useful.